program research experience for undergraduates.
Okay, but not just classes.
We have classes, and we'll get into that during the tour.
Okay.
Yeah, thank you.
Thanks, Katie. So today I'm going to talk to you about the future of food from the sea in a changing environment. Before I get into that, I'll go over a little bit of my background. So I got my first exposure to the seafood industry when I was 14. I started working at a local seafood restaurant in my hometown in Scarborough, Maine, and I loved it there. I loved working there. I kept it up throughout all of high school and college as much as I could. And so I went to Bowdoin College, where I majored in neuroscience, but I took a lot of classes in earth and oceanographic science, and I found myself every summer looking for a research internship that would be on the ocean or studying marine species. And so from there, I went on to get my master's in environmental toxicology at New York University, where I looked at the effects of contaminants in the Hudson River on sturgeon larvae, which are these fish pictured here. And so from there, I came right to USC, where I started the PhD program in marine biology. I'm currently in my third year here at USC, and I'm studying the physiological responses of marine invertebrates, specifically urchins, to environmental change. And so one of the overall goals that this research can be used for is to be able to increase the efficiency and sustainability of food from the sea. And so thinking about food from the sea, so the oceans cover over 70% of our Earth's surface, and they make up about 97% of the Earth's biovolume when you consider how deep the oceans are. And so this offers us a substantial capacity to meet humans' need for food. And so looking at our humans' population, you can see that we've been growing exponentially. And so today our population is about 7.7 billion, but it's projected to reach 9 billion by 2050. So in the next 30 years, we're going to have over 1 billion more people on this planet that we're going to need to be able to feed. And so that's where the ocean can be a valuable resource for us to meet these needs. And so over the course of human history, humans have become great fishermen. So we've had recent developments in technology with boats, with sonar systems, with different fish tracking abilities, and we've become so good at fishing that we've actually depleted most of the ocean's big fish. And so with this huge decline of the ocean's big fish, it's creating an even bigger gap between our increasing demand and our decreasing supply. And so in order to bridge this gap, one possible solution is going to be with the aquaculture industry. And so aquaculture is the growth of aquatic species, both saltwater and freshwater, and it includes fish, shellfish, algae, etc., anything grown in water. And so here you can see on this graph that our wild capture production has actually plateaued or become relatively stabilized over the last 30 years. And that's in part due to the decline of big fish and also in part due to newer regulations to prevent overfishing. But since we do have this increasing demand, you can see that aquaculture has actually begun to pick up that slack and has really increased over 30 years. And so looking at aquaculture in the U.S., the top aquaculture species in the U.S. is oysters. And so they produce about 37 million pounds annually, which equates to $192 million worth of production. And then oysters are followed by clams, and then salmon, and then mussels and shrimp. However, 90% of the U.S. seafood consumed is imported. So this means that U.S. aquaculture farmers are only meeting less than 10, or about 10% of our country's demand for seafood. And on top of that, most of the aquaculture in the U.S. is actually freshwater. And so our marine U.S. farmers are actually supplying only 2% or less than 2% of our demand for food from the sea. And so in order for us to not have to rely on other countries, which we don't have to want to be dependent on them in order to be able to make our own food, we're going to need to, or aquaculture offers us a big opportunity to be able to increase. And so why are oysters a good species for aquaculture? So one of the major reasons is because they're herbivores, or they're lower trophic species, so they're low on the food chain, which makes them have a much smaller environmental footprint. So if you think of if you're trying to grow a really big fish, you would first need to grow really small fish or capture really small fish from the ocean, and then maybe a medium-sized fish so the small fish can eat the medium-sized fish, then the medium-sized fish eat the large fish. So you have to find or capture medium and small-sized fish in order to be able to produce your big fish. So that wastes a lot of energy. So by using oysters, which are low on the food chain, they can filter their own food through algae in the ocean, so they don't need to be fed. So this gives them a much smaller environmental footprint for growing them. And on top of that, since they do filter algae naturally in the ocean, they're also really good at cleaning the water. So as you can see from this image, if you put oysters into a tank of water, that they're able to clean it out, which actually helps increase biodiversity in the areas that they live in. So they're good for the oceans in that sense as well. And they're also a good source of protein, so they're considered a healthy food for us to eat. And so looking at the oyster life cycle, like most marine animals, oysters have a complex early life history stage as larvae in the ocean. So similar to how butterflies have a larval stage as a caterpillar, frogs have a larval stage as a tadpole, oysters also have a larval stage where they're free-swimming microscopic plankton in the ocean. And so oysters reproduce. So an adult male and female will, under the right temperature conditions during the summer, will release their gametes into the ocean. And a single female can release over 100 million eggs at once. But the problem is that less than 1% of these will survive into adulthood. And so from the time the egg is fertilized, it takes oysters less than 48 hours to build their shells. So they can build their shells within 40 hours where they become into this stage where they're about the diameter of a human hair. And they'll remain in this larval stage, again, as they're free-swimming plankton in the ocean, vulnerable to, the tides and everything else in the ocean. And then they'll grow this foot after about 3 weeks, and they'll find something to settle onto, preferably adult oyster shells. And then from there, once they settle, they're immobile, and they'll just continue to grow there. And it takes them about 2 years to reach reproductive maturity where they can be. And so how do we raise oysters for the aquaculture industry? So we start them off in a hatchery. So this is actually, we have one of these here on Catalina in this building. It's on the 3rd floor. And so thanks to the Wrigley Institute resources, we're able to have relatively large-scale cultures for our needs. So these are 200-liter vessels, and each one of them can hold about 2 million larvae. And so from here is where we'll grow them up until they reach metamorphosis, or until they settle. And then we're fortunate enough to have Carlsbad Aqua Farm take care of them from there for us. So we'll send them over to Carlsbad. It's just south of Los Angeles. And they'll go into the nursery phase at this point. And so they get put into these tubes that then there's seawater with algae upwelling through them so that they get fed until they grow to this big enough size where they can be put into cages. And then this is called the grow out phase, where these cages are then attached onto these buoys, and they'll spend the rest of their days. And you can see here a picture of, so these are oysters that we spawned here in this building last summer. And you can see they're already a good size, so they're almost able to reproduce again this summer for us after just 1 year. But that's not to say that there isn't a lot of challenges for the aquaculture industry. So for example, temperature. So the oceans have been heating up pretty rapidly in recent years. So it's going to be important for organisms to be able to adapt to this increase in temperature. And then other stresses in some areas, ocean acidification is already becoming a really big problem for growing, particularly organisms to calcify or build a shell. And oxygen depletion and pollutants can also be big issues for the aquaculture.
Question for you?
Yeah.
How long do oysters live?
So they can live over 30 years. Oh. Yeah, depending on the species.
And how old are they when they harvest them?
So about 2 years
3
is
years.
typical. Yeah. Okay. And so these early stages of development, when they're in the larval stage, are when they're most susceptible to these environmental stresses. So that's the stage that we choose to study in our lab. And so in order to look at how these environmental changes affect organisms, there are two major influences, being nature and nurture. So nature is the effect of is due to genetics. So basically what genes you're born with, how that affects your appearance and your personality. So just like in humans, where if you come from a tall family, you're more likely to be tall. If your parents are tall, if you're shorter, if your parents are shorter, you're probably going to be shorter. And so the same thing kind of holds true for larvae. So you can see that through this image, larval family. So each of these bars is a different larval family. So the larvae came from the same mom and dad at the same time. And you can see that some of them have growth rates of less than 4 microns per day, and some can have growth rates of over 14 microns per day when they were all raised in the same lab conditions, so that at the same temperature, given the same food in the same room, and we see these differences. So these differences are likely due to their genetics. And then on the other hand, we have the influence of nurture. And so this is due to what environment you grow up in. So if you have identical twins that have the same genes, and one of them grows up in Alaska and the other grows up in Florida, the one from Alaska is probably going to be more cold tolerant. And so that's where physiology comes in. And you have changes over a shorter time period, so within your lifespan. And so how we study these processes here on Catalina. So here we're mostly interested in these physiological rates, which allow us to basically ask the organism how it's doing and figure out whether or not it's stressed out in its environment. So here are some of the ways we can do that. First, we can measure its growth. So here's an example of how we would measure shell length for oyster larvae under the microscope. And so measuring their increase in length over time can give us a growth rate for them. And then we measure their total protein and lipid body composition, so we can see what their biochemical constituents are or what they're made up of. And then in terms of rates, so we measure their metabolic rate or respiration. So we can put hundreds of larvae into these small vials that have a sensor spot on them, which allow us to optically quantify the amount of oxygen in this vial. And so by measuring the oxygen depletion over time, it gives us a respiration rate, which then we can convert to know how much total energy an organism. And another process that we're particularly interested in is protein synthesis rates. So this is the most energy-consuming process of the organism. And so we do this we use radio-labeled amino acids, which amino acids make up proteins and are known as the building blocks of life. So proteins are required for almost everything. And so we're able to calculate these rates over time. And then lastly, I'll just talk about feeding rates. So if we give a known amount of larvae, a known amount of algae, we have a particle counter which can monitor the algal concentration. So by looking at the decrease in algae concentration over time in the samples, we know how much these larvae are eating. And so our overall goal with all of these different processes is to be able to look at the mechanisms that larvae are using in response to different environmental conditions. And so one way that we can use to depict this is through these energy budgets. So this can be shown through a pie chart, where the size of the pie is going to be the energy input, so the amount of total energy that the organism has. And then we can allocate that off into different processes. So like I said, protein synthesis is the most energy-consuming process that an organism has, and it requires over half of the larva's energy budget. So that's like if every other breath you take is going towards synthesizing protein. And so one of the major findings that we found in this lab is that when we increase the temperature, there's a disproportionate increase in the amount of energy that larvae are getting and the amount that they need to use. So here, under this increased temperature condition, you can see the pie increases, so they have more total energy. But you can see that the amount that's going towards protein synthesis is even more increased. So larvae need to spend more of their energy on synthesizing these proteins. And so maybe you can start to see how this could be problematic, because in this scenario, this gray shaded area, there's a lot of extra energy. So this extra energy needs to go to other processes that are vital for life. And then also some of it can be used as a reserve energy for if a larvae encounters another type of stress, like a virus or bacteria or ocean acidification or something else that it needs extra energy to deal with. And so once you increase that temperature, that amount of excess energy is getting lower. So larvae are going to be more vulnerable to stress and may not have enough energy to deal with it, which could cause mortality. And so in addition to these physiological energy allocations, we're also interested in the effect of genotype. So I'll go over one more example, looking at different larval families so we can get at this effect of nature as well. And so in this example, for these results, I'm looking at 3 different larval families. And you can see they all have a similar growth rate, so about 6 or 7 microns per day. So these 3 families were grown in the same environmental conditions, same temperature, given the same amount of food, and they have the same amount of, and they're growing the same amount. But when you look at their feeding rates, you can see that there's a 5-fold difference in feeding rate. So in this case, family 12 is eating 5 times as much food as family 40, but they're growing the same amount. So a human analogy would be like if you could eat 5 hamburgers and your friend only eats 1, but you're both gaining the same amount of weight from that. So yeah, that's what we look for. That's what would be a good thing for us as humans. And so we're looking, so basically the overall goal of our research is to be able to pick out what makes a larvae most efficient, which ones are fast growers or slow growers, and look that are responsible for that, so we can ultimately select to breed those oysters for future generations.
They taste the same if they grow fast or just...
That's another thing to look at, yeah. And so just to conclude, so the concept of domestication in farm species for human civilization has been going on for a really long time. So humans have been cultivating corn for thousands of years. And this led to the Green Revolution in the 1950s and 60s, which was a huge increase in the amount of agriculture production due to purposeful selection of traits and creating fast-growing hybrids. So now in the present day, we're looking to further this into the Blue Revolution, where we can apply the same techniques for the aquaculture system, with the ultimate goal being to be able to manipulate oysters to create fast-growing hybrids and make them able to be resilient to environmental challenges in the future and even soon.
I have a question on the protein.
Okay. So that was the technical lab. So I'd like to thank the Manahan Lab and all of its members for their help with all of this work, the USC Wrigley Institute staff for all of their support, and thank you all for coming. Yeah.
The temperature where the protein increases protein development, I mean, you say that it doesn't have as much energy, but wouldn't the protein development be because there's available energy, and if there's a stress, it might just stop producing the proteins and use the other energy for a stress situation?
So it's a very complicated dynamic. Yeah, and so the thing is, and that's a good point. So one of the things that we're actually interested in looking at is the depositional efficiency, because like you said, so a lot of times you synthesize protein, but that doesn't necessarily mean you're adding protein onto your body mass. There's a lot of protein turnover and protein degradation that's going on. So
so it could be having to do some more protein because the heat is making.
Yeah, exactly. So the heat could be degrading the protein, so they need to synthesize more. So
Is it the same for the different genomes with the fast growing?
So that's something that we're looking at right now. Yeah, and so we do see there's different effects with different families.
Why oysters?
Why oysters? Yeah, so oysters because they're a lower trophic species, so they can eat algae. Also, they don't need to be fed. You can grow them really crowded together. They don't need any space to move. And they have a relatively short generation time, so they can be ready for market in 1 to 2 years versus bigger things.
Are they more susceptible to pollution because of their filters?
So it's tough to say. I don't know. So I know that sometimes it becomes a problem because the oysters will maintain, so if they're filtering through these pollutants, they'll add them onto their body, but also due to like bioaccumulation and biomagnification, it actually could be more of a problem for higher trophic species. Like that's why they say it's dangerous sometimes to eat tuna because of the built up over.
There another product besides just oysters that you make out of them? I'm allergic to oysters, so I'm hoping your research doesn't go too fast.
Yeah.
but oyster, I mean, do they make like dog foods and cat food?
Yeah, they could definitely be a good fish feed as well, or dog food. Yeah, it's I think it would be very helpful.
They're just not grown.
It's not just humans, yeah.
Just not cultivated for human consumption.
Yeah,
Okay.
yeah. I think people are working on making like protein powders out of
Okay.
oysters.
You go first.
The aqua farms, when oysters are grown, do they require artificial source of food, or do they just naturally grow?
No, so
in the
yeah,
water?
so once they get in that grow out phase in the ocean, typically I don't think they're fed extra. Yeah, they can just use the algae that's natural.
And is it so the environment where they grow, related to the question about pollution, is it controlled? The water where they sit for a year or two?
Yeah. So I think that the locations for the farms are particularly chosen to be areas that would be free from that pollution, that type of pollutants.
The one in Carlsbad, is that in some of the lagoons there?
Yeah,
okay,
exactly.
So, first, thanks for a really great talk. So I was you made me think when you showed the species that we could rely on more in the future were heavily these shellfish species, which have this bad reputation of having higher risk of bacteria or disease that goes all the way back to the kosher laws, from thousands and thousands of years ago, and then pregnant women are advised against eating shellfish because of the risk of contamination. And so how do we reconcile that if we want to scale this up?
Yeah.
Is there some scaled up risk that we're encountering then?
Yeah, so, and I think a lot of the risk is particularly due to eating them raw. So by cooking them, you remove a lot of that risk of like bacteria and viruses in that way, because you can kill those off before you eat it. And also, they're getting better with technology to be able to look at, so paralytic shellfish poisoning is another big issue, but with the increased technology, they're able to tell pretty much right away, I think now, or they're able to test for that, whether that may be a problem, and then maybe there's a season where that area can't sell any of their seafood. But
Thank you.
yeah, it's another thing to be worried about.
You mentioned paralytic shell poisoning?
Yeah, so it comes from, so sometimes toxic algae can bloom, and then as the oysters are filtering that toxic algae, it can become paralytic for a few reasons.
Is there any advancement in the use of the shells? I mean, what happened? It's got a lot of waste product.
Yeah, so the shells. Yeah, so that's one thing. So the industry actually, so oysters larvae prefer to settle on adult shells. So that's one way to kind of help more larvae survive, is to smash up the shells into what's called culch. So it's mashed up adult oyster shells, and spreading that into the water will help the larvae be able to find those little pieces of shell to settle on. So we use those in the lab to be able to help grow more oysters.
Did they have any idea why they like their same species shells?
Yeah, so it's thought that
Almost parasitic.
yeah, so it's thought that because that means that particular oyster, that was a successful place for someone else. So if it was a successful place for another oyster, it would probably be more likely to be a successful place for this one. So there's different ideas as to why, but.
Able to filter out CO2, and you're gonna.
Yeah.
Questions? Thank you so much.